All right, good morning, everyone, and welcome to our webinar this morning on sleep. We are very fortunate to have Dr. Shu Buckley with us this morning. Dr. Buckley is the director of the Sleep Disorders and Neurodevelopment Consult Service at the National Institute of Mental Health. And Dr. Buckley, thank you so much for joining us this morning, and we're very excited to hear from you today. And welcome and take it away.
Thank you. Thank you very much. Good morning, everyone. Thanks for joining. I know you heard from my colleague a couple of weeks ago, and that it's really great to be able to split this talk with two people. So we're very happy to have Dr. Concepcion Zayas with us. And so today I'm going to do a little bit of a recap, but not a whole lot. And I want to kind of focus on the work that we've been doing at the Sleep and Development Core really for a couple of decades. So the service as it exists right now started probably maybe 6 or 7 years ago. For a very long time, we've been putting together the bones of what we need in order to study sleep comprehensively and understand how it impacts the developing brain and body. So before I get started, I always want to thank the friends, the children, the families, the people who participate in our research, without whom we have nothing to say. And to my incredible staff. So running a sleep lab requires 24/7 effort. So we have some wonderful daytime people, some wonderful nighttime people, and just a really nice cohesive team that makes this all work. So thank you to them especially. So I want to start with some sleep terminology. And you heard some of this a couple of weeks ago. So I just want to ground you when you hear me saying certain terms, what I'm referring to. So sleep architecture can be macro, and that's sort of the terms that you're used to hearing. How much time did you spend in stage 1? How long were you awake? How long did it take you to get to dream states? Those are latencies. Then there are also these micro architectural facets to sleep. And that's really what we study, what we're contributing to the field, because this is very understudied. And I'll show you what those are as the lecture goes on. So you'll hear me talk about macro and micro architecture. Ultradian rhythms, I'll show you an example of what that looks like. This is the nocturnal cycle of sleep stages. So when you go to sleep, you just don't turn off your brain. You're actually sort of cycling in and out. I'm sure you all know this: light sleep, deeper sleep, and then dream sleep. And you do that a number of different times a night, and those are called ultradian rhythms. Circadian rhythms, on the other hand, are something that we're all familiar with. That's the 24-hour rhythm of sleep and wakefulness. And the important thing to understand about circadian rhythms is that they don't just impact the brain. The brain and body need to be aligned, and organs have their own circadian rhythmicity that gets coordinated through the brain and the suprachiasmatic nucleus. So this is an interesting and complicated area of study that we're not actually going to touch a lot on today, but just so you understand what that part is. And sleep regulation. How is sleep regulated? We'll do a quick recap of the processes that we define regulation by, and sleep patterns. So when someone says sleep patterns, they mean all of these things, or one of these things, or a couple of these things. And so it's always good to orient yourself when people are talking about sleep patterns. Which of these are they talking about? What are they focused on? So sleep, the simplest definition is from Dement, grandfather of sleep medicine, and it's this reversible behavioral state. There's a disengagement from the environment, and you're more or less unresponsive to it. And so that's the behavior. And so when people say, my child doesn't sleep a lot, or they're up in the night, they're really commenting on the behavior of sleep, which can be characterized in lots of different ways. And as a child neurologist, a developmental person, sleep has always held an incredible pull for me, because it is the primary activity of the brain during the earliest parts of development. And so when I started my career in child neurology many years ago now, I wanted to understand better about what's happening during this period of time. These are Golgi plate sections from the frontal cortex from Connell's Atlas for a 1-month-old and then a 4-month-old. And you can see the massive amount of change and connectivity in these pyramidal cells and these cells in the brain. And as the brain is growing and restructuring and scaffolding itself, those changes are reflected throughout development in the sleep profile. And so this is the profile of infants through adult age and sort of breaking up that macro architecture that we talked about. How much time do you spend in REM? How much time do you spend in non-REM? And you can see through the first 5 years of life, there's this massive front loading of REM sleep. And then your sleep organization changes as you get older. And I always thought that that was really interesting. What's happening during this period? What is sleep doing? And that kind of brings us to the second part of sleep, right? So it's a behavior, but then it's a function or functions. There are many different functions of sleep. The best studied is the role of learning and memory. Strongest evidence points to sleep's primary function as a regulator of brain plasticity and cognition. And again, from a child neurology and a developmental perspective, that's really fascinating, right? Likely REM and non-REM, they have differential roles in the developing brain versus the mature brain versus the aging brain. This memory consolidation part of this neuroplasticity part of sleep is the primary function of sleep as we understand it. And there are many more sort of other exciting sleep functions, right? There's a very recent sleep function proposal that is very relevant to neurodegenerative disorders and dementias. And that has to do with the glial cells being used during slow wave sleep to clear debris. So that slow wave sleep then has a function in older age to sort of get those toxins or those tangles or tau proteins or whatever it is that's accumulating and causing dementia cleared from the brain. So it's a fascinating area of research. This synaptic plasticity part is what I was focused on when I first started this research, and that's the primarily best understood function of sleep. So we have a behavior, we have a function, and then we have a process. And so again, I'm just going to repeat those things throughout this lecture. Sleep is a behavior, it's a function, functions. There's also a process, and it's the process part that allows us to start digging deeper and trying to understand better about what goes wrong in that process in children who have neurodevelopmental disorders and people who have psychiatric illnesses. So I know you covered this last time. I'm just going to do a very quick reminder that we think about sleep being governed by this two-process model. There's homeostasis and there's the circadian rhythm. And so these things are the sort of the yin and yang, and they are complementary. So for example, when you're awake, the pressure to sleep rises during your wake time. It reaches a certain threshold, probably governed by different things that we call somnogens that accumulate in the brain and really bring the pressure on to go to sleep. And so then here's sleep time. This is 11 p.m. at night. And then quickly that homeostatic pressure, you can see it kind of just quickly in the first third of the night dissipates. And what keeps you awake at that point is the yang, or the circadian pressure. So this is governed by melatonin. And so you have, before you go to sleep, a rise in melatonin that keeps you asleep during the sleep period after your homeostatic pressure has dissipated. And in the healthy brain, someone who is aligned with both of these processes, these are very nicely lined up so that the homeostatic pressure is completely dissipated as melatonin drops, and now you're awake and ready for the rest of the day. So that's the conceptualization of the process. There's this other process part of sleep that I've been very interested in, and that's the electrophysiology specifically. And so this brings us to how do we measure and analyze sleep disturbance? So we know sleep is very important, and it kind of gets in the background of sort of general essential things that you need for health, but it really should be front and center, along with access to fresh and healthy food, access to shelter, right? Access to anything that supports your life, fresh water. Sleep really is that important. And so we try to measure both the quality and quantity of sleep, and most of the measurements that are out there are subjective, right? Meaning we ask you questions, you tell us, or your parent tells us about how that goes. We try to get closer to objective measurements with actigraphy, and this is a proxy for sleep-wake, right? If you're moving and, then we think that you're awake. This is very simplified. And if you're not moving, then you're asleep. But what we've been very interested in our lab is integrating the sleeping physiology, because there's a lot going on during sleep, both in the musculature, in respiratory parameters, and in the brain. And here's our little friend of ours hooked up for the somnogram in our lab. And what we're very interested in is this oscillatory information. So these neural signatures in the brain and how they relate to the rest of the sleeping body. So that's how we measure it. This is a little girl with our gold standard. These are actually gold leads that are taped on, glued on, taped on, and then we record. And the EEG, so these scalp leads, these gold leads at the top here, reflect the postsynaptic activity of 100 million or a billion neurons. It's an enormous amount of information. And so that's the other thing I want to impress upon you. It's so much information. And when we record from the sleeping brain, we can take those waves, that information, and they can be further divided into sleep states. And there are differences in frequency. There's differences in amplitude. You see the huge amplitude here for slow waves, and differences in morphology. So this is like a lesson in decoding. And this is also what I ended up falling in love with when I was a student, that you can watch the sleeping brain in real time. And just by being able to decode these things, I can know we're in light sleep, we're in deeper sleep. Now this person is very deeply asleep. And these little signatures, this one in particular, we're going to spend a little time on, is called the spindle. And it has been associated to cognition, learning, behavior, basically the health of a very important microcircuit in the brain. And so it's an assay of health on a lot of levels. So we take those signatures and we decode them. And the way that you read a sleep EEG is 30 seconds by 30 seconds. So you can imagine when you go into a sleep lab, done correctly, you have somebody looking at the 8 hours of sleep, 30 seconds by 30 seconds, and determining by those little keys that I just showed you, whether or not I'm awake, I'm in REM sleep, I'm in 1, 2, 3 deep sleep. And this is called a hypnogram, back to this term ultradian rhythm, right? How many times did we go in and out of this? Is it distributed in the way that I know the sleep process is supposed to go? In this instance, this is a very healthy, cartoon of what an ultradian rhythm or a hypnogram would look like. Very heavy distribution of this purple, which is slow wave sleep in the first third of the night. And we talked about that, right? The dissipation of the homeostatic pressure. And then longer and longer cycles of REM sleep. And you can see here the red on top toward the last third of the night. So homeostatic pressure is dissipating. Now we're into REM sleep. And these different sleep states likely have different functions, especially in the developing brain. And so it becomes important what order and what this looks like. So when we look at different populations with different neurodevelopmental disorders, we're looking for these things. Do we go into REM sleep? Do we have a normal ultradian rhythm? Is there any slow wave sleep at all? These are all clues about what's going on underneath the scalp. And what's important about that is that the rhythms and the decoding allows us to stage, but what it's really telling us is what's happening underneath. And each state has its own neurotransmitter bath or milieu, if you will. Each state has a different neurotransmitter combination. So for instance, in the awake state, you're going to have a lot of acetylcholine, you'll have the SSRIs, right? Not the SSRIs, I'm sorry, you'll have serotonin here, norepinephrine, and then orexin. In non-REM sleep, there is a different combination. In REM sleep, yet another combination. And so just sort of to make this more concrete, the loss of this neurotransmitter in the brain that we think is secondary to an autoimmune insult is what results in narcolepsy. So and that was just discovered maybe 20 years ago now, 20, 25 years ago now, right? So as we learn more about what these signals mean, about what guides them, we can walk them back into mechanism and into pathology and try to figure out what's actually going wrong in the brain. So before 20 years ago, we knew what narcolepsy looked like. We didn't know what caused it. It's actually the absence of this particular neurotransmitter, because it turns out that orexin is a stabilizer between all of these states. So it's just an interesting little aside fact. And that kind of brings us to what we've been doing. So sleep is a behavior, it's a function, and has many functions, and it's a process. And if you can define the process, like learn the code, then when you deviate from that code, you can try to walk that back to figure out what's going wrong with the structure of the brain. And because it is so well mapped out, that gives us an entry point. So I want to come back to this two process model of sleep. Like everything, that is a very simplified and easy way to think about it, right? Here's process S and here's process C. But in real life, there are many other things that can unyoke or change that direction in one way or the other. So you all know children who don't go to sleep at 8 o'clock, even though probably their homeostatic pressure is telling them they need to, right? Or the flip of that, which would be adolescence, when you get to, right around puberty, your homeostatic rate changes. And so it starts to actually take longer to get to that state where that pressure is actually telling your brain to go to sleep. But we don't change our cultural norms around that, right? So now you're not a primary schooler. Now your physiology has changed and your slope has changed, and you're going to take you longer. It's not going to be 9 or 10 o'clock. It's going to be more like 12 or 1 o'clock when you feel that homeostatic pressure. But then we still ask you to get up at 6 o'clock and catch the bus in the morning. And so there are all these other determinants that are likely much more important when we study sleep, right? Out of sort of the lab, but in real life, that have a much bigger determinant on what this balance actually looks like. And one of those things is genetics. And when I say genetics, I don't just mean a disorder that we can firmly say can be tracked back to particular genetic mutation. I mean the combination of genetics that make us who we are. And so we want to sort of figure out, how do we incorporate all these other things to help us better interpret this process? We think that sleep metrics, and in particular, some of the metrics I'm going to talk briefly about today, are a way at least to start to get there and open up that conversation. So here is a really nice figure that was published a couple of years ago now, and I always use in talks because I think it's very clear what it's saying. And this says ASD, so short, the new way that we conceptualize autisms, right? ASD. But it could say schizophrenia, it could say anxiety, it could say depression, because sleep disorders and disruption are ubiquitous in mental health and neurodevelopmental disorders. It's just part of the fabric. And that's kind of what this figure is sort of proposing here, that there are two ways to look at sleep disorders, that you have the initial disorder, and then that leads to behavioral dysregulation. And because you're behaviorally dysregulated, you dysregulate your sleep, and then you get into this vicious cycle. Or the disorder itself, baked into the disorder, is a change in behavior and a change in sleep. And the sleep can be a behavioral change, or it can be a change in process that's not immediately apparent. But either way, those things end up changing the function of sleep. And that's really what we're interested in. If sleep is supposed to be supporting and developing the brain, when there are sleep disruptions, we end up with an outcome that's not optimal. So the field is starting to think much more like this, that disorders that have an increased prevalence of sleep disruption are actually intrinsically the same thing that is causing the behavioral disorder is causing this regulated sleep. And that gives us a nice window into what we might be able to do with that. So I'm just going to step back and that was a lot of information, and just tell you that, yes, it's a lot of information, and it's very hard to sort of integrate and weave together. And we've been working at this for a long time. And we realized that in order to move forward with our sleep research, we need to do a bunch of things. And the first thing that we needed to do, we did about a decade ago now, which is to convene experts in the field at these sleep and neurodevelopment workshops that are supported by NIMH on campus in Bethesda, to get people together and to say, what is it that we don't know about how sleep builds the brain, about how sleep during childhood optimizes outcomes, or disrupted sleep causes a decrease in quality of life. And so we had to identify data gaps, followed a couple of years later by infrastructure. I'll tell you a little bit what I mean about that, which is just getting everybody on the same page about what needs to be collected and explored and shared in order for us to move the research forward. We had some nice funding from the Pediatric Epilepsy Research Foundation to bring a bunch of groups together and start to build that infrastructure so that we're all speaking the same language at different pediatric centers of excellence. And then a couple of years later, followed by a workshop thinking about how do we translate the information that we're getting in foundational or basic science into the clinic and then into the population. And you can kind of see over the last 5 years of data, since we've been holding this increasing interest in sleep and neurodevelopmental disorders, at least as measured by the number of publications. And so that's definitely gratifying for us. And here's what I was talking about building the infrastructure. So what we did is we said, there are a lot of these centers of pediatric excellence that do sleep studies that record this oscillatory data from the brain. And then what do they do with that? And so we partnered with the National Sleep Research Resource. And here it is right here. And for anyone who's interested, there are many data sets, now many more pediatric data sets. And here's the URL. You can go and take a look and play around on this site. But we got these data sets off the shelves into this repository that is designed to ask and pull out information. I'll show you a bit about what that might look like. And our most recent partner is Northwestern. And so we start to sort of make sure that everyone's speaking the same language so that efforts aren't done in silos and then everyone is sort of not knowing what's going on behind closed doors. So that was a big part of what we've been doing. Here's an example about what this might look like. This looks like a busy slide. It's not really. This is from Nationwide Children's Hospital. It turns out that this major center, children's center, had acquired many thousands of sleep studies in the same way, standardized for the same thing. Children who came in maybe for snoring, or they came in because they had trouble sleeping, or whatever it was. They decided that they were going to deposit those 3,400 plus sleep studies into NSR. And that allows us to go in to look at very particular areas in the brain. These are just 2 leads in the frontal part of the brain, so frontal, and 2 in the central, and take a look and see, can we now begin to construct developmental trajectories about what age does to these parameters that we're looking at. These are the libraries that then we were thinking that we needed in 2014 when we said, hey, we have a data gap here. Before we can actually definitively say that this deviation is particular to this disorder, we need to have roadmaps and libraries about what it looks like in typically developing children. So here's just a little snapshot of this, right? So we can say stage 1, stage 2. So the code for these things, this is where spindles occur, right? Stage 3, this is slow wave sleep, REM, or rapid eye movement or dream state. And we can start to say from 0 to 20, this is what that curve looks like. This is what it looks like from 0 to 20 for stage 2. This is what it looks like for slow wave sleep from 0 to 20. And this is actually a very nice check against literature that's already out there that we already know that as children go into puberty and then into young adulthood and then into adulthood, the duration of slow wave sleep and even the magnitude changes. So you can look at duration, you can look at percent. So what percent of the night are we spending, excuse me, in any of these states? So this is the power of large data sets. And I want to just, excuse me, point that out, that once you start getting thousands of studies of the sleeping brain, we can start to build these libraries. And age is hugely important factor in what any of these things look like. And you can go a little deeper. So we talked about macro architecture, right? So I'm going to go back a slide. REM sleep, the percent of REM sleep, the percent of slow wave sleep. These are macro architectural details. Then we can go deeper into things that reflect the process part of sleep that we can walk back into function, but that you might not ever see in behavior, right? So an example would be that little morphologic thing I told you that says you're in stage 2. That's called a spindle. I can say, I want to pull out spindles. I just want to look at spindles. And I want to look at the fastest ones because they have a certain frequency that they oscillate at. And you can say, I want to know, what does fast spindle developmental trajectory look like through 0 to 18? And here it is, right? And once you do that, then you can say, well, I want to know what fast spindle trajectory looks like between 5 and 10 for people with Down syndrome, for people with ASD. So you see, you can start to disentangle different diagnostic groups, differentiate them on these parameters. And that's a really powerful thing to be able to do. Slow oscillations, the reason I have these up here is that any one of these signatures doesn't necessarily tell you the whole story. But the way that these signatures, these different oscillations interact can tell you a whole lot more. So for instance, the relationship between spindles and slow oscillations has a lot to do with memory and learning. And so these are just examples of what you can do with this information. And so, of course, the overarching goal has always been to better leverage this information in the many patterns of sleep. And we talked about the terminology, and that pattern means sort of all of these things put together or some combination of them that will allow us to identify deviations from that typical developmental trajectory prior to there being suffering. So prior to clinically aberrant behavior, right? So one of the biggest problems that we have in mental health research is that because we rely so heavily on behavioral presentation, the time that we are addressing the mental health malady, person is suffering, has been suffering, and maybe has already lost function, right? So it's kind of like we have these regimens for folks who have high blood pressure to prevent the stroke, right? We have barometers for people who maybe have blockage in their arteries to prevent the heart attack. So this line of thinking is there a way to leverage what we know about the way the brain builds itself during sleep that we can identify and intervene prior to there being the heart attack or stroke, right? Prior to the mental health crisis. So that's the idea behind this. And I just want to impress upon folks again that this is a diagram that is modified from the Bible of Sleep Medicine, which is Principles and Practice of Sleep Medicine. I think Dement was one of the editors here, and this underneath here it says bipolar disorder, but it could say any behavioral disorder. Could be autism, could be schizophrenia. The overlap with psychiatric disorders and sleep disruption is enormous. In this case, it's insomnia. But what I want you to just get from this diagram is that functional impairment can take lots of different pathways, and a lot of those pathways lead back to sleep disruption. So we can take it from the top. We can say there's this behavioral disorder. You have a child, they're presenting with what looks like extreme anxiety and or bipolar disorder. They have insomnia. That insomnia or sleep restriction results in emotional regulation deficits, changes in affect, right? That's functionally impairing. Same thing, you can go down the cognitive route, changes in cognition, you're sleep restricted, functional impairment. Systemic health, you have increase in inflammatory markers. Now you have global health dysfunction, functional impairment. So the burden of sleep disruption in psychiatric diagnoses and neurodevelopmental diagnoses really mandates that we take a deeper look at sleep and what's happening. Here's an example of what I'm talking about, that sleep is intimately related to mental health. This is a 2019 study of 376 children from 11 to 18. They completed measures of insomnia and sleep behavior. And the take home from this paper is that 75% had mental health diagnoses. Most significantly disrupted were people with affective disorders, so mood dysregulation and ADHD. So a major tenet of sleep and neurodevelopment research is that early sleep patterns can be predictive of neurodevelopmental outcomes. And again, that terminology pattern encompasses all the different things that we talked about, right? The behavior, the oscillatory measures. We can figure out the algorithm about how these patterns fit together. Hopefully we can intervene. So most of the research, and certainly everything that I showed you in the last 5 minutes, is retrospective. So it means we already collected, it's already sitting on a shelf, we're taking it, we're looking at it, we're trying to figure out hypotheses from data that's been collected for another reason. So what we started at the NIH a couple of years ago now was a prospective study, and this is ESP, or the electrophysiologic sleep phenotyping study. So this is a study where we're taking children specifically for this reason and looking at them and looking at their sleep prior to any development of any psychiatric illness, trying to continue to build on that library about what do developmental trajectories look like in children who either do not have a flag for developmental delay or problems, or children who might. So we have two very broad categories: children who perhaps have a language delay, they have a parent with schizophrenia, they have been flagged for some other developmental maybe lag. So we're looking at those two groups of kids, we're bringing them in and we're marrying those EEG oscillatory phenomenon that we talked about with what do they look like behaviorally. We'll do genetics, we'll look at psychosocial and lifestyle data. And then we'll follow these children, each one, depending on when they came in, either 4 times at the NIH overnight or 3 times. So the younger the child comes in, the more rapidly their brain is changing, the more times that we look at them. You come in a little bit older, 5, 6, 7, 5 and 6, we see you 3 times. And so this is a really powerful construct. It takes a long time, but we're able to do that at the IRP. That's one of the benefits of working on campus at the NIH. We're able to do these longitudinal studies where we are fully immersed in each child. And I'm going to point out these 2 wonderful Spideys here. Both of these little boys are 6. They live in very similar neighborhoods in the DMV. They both have a parent with schizophrenia. One child behaviorally has no flags. The other child has what looks like intellectual disability and some other affective issues. So what are the differences in objective sleep oscillatory biomarkers as we follow these children that will help us understand their differing trajectories given very similar genetics? So this is what it looks like to come in and be a part of ESP. It's a less than 24-hour admit. You come in, you get a history and physical and a neurologic exam. You do a couple of questionnaires, and then we look at the sleeping and the awake brain. You get a neurodevelopmental history and assessment, very streamlined. If it looks like that there are concerns for autism, you get an autism screening. And then very minimal blood work just on the first day. So we need to look at iron and CBC to make sure that those things are healthy when we're looking at sleep. But the other very wonderful part of this is that we partnered with NIAID, which is the Infectious Disease and Allergy Institute, for a centralized sequencing protocol. And so what this does is there is a group at the NIH who will work with the families, especially if you have concerns about developmental delay or intellectual disability or any type of genetic concern, and they will return the results to you with a fully certified clinical geneticist, and they have been a wonderful part of our evaluation. Okay, so one thing that we didn't talk a lot about today, but that you did hear from my colleague a couple of weeks ago, is that so overarching all of these other things about sort of understanding sleep and what sleep is, right? A behavior, function, and process, are then the primary sleep disorders. And so the most prevalent primary sleep disorder that we see a lot and that people talk about is obstructive sleep apnea. And so children with obstructive sleep apnea, for the same reasons that we just talked about, all the things that sleep disruption for insomnia does, they can have those things too, right? Change in affect, change in cognition, health issues, particularly cardiovascular issues, because every time you chop open that airway, you're causing stress on the system. So there's all sorts of reasons why we don't like OSA in childhood. It turns out that children with OSA also have an increased risk of depression. So it's one of these things, the more you know, the less you know, and you have to keep looking and trying to figure it out. And so here's a really nice study that looked at 450 children, and they had OSA. And so they said, okay, half these kids are going to get tonsillectomy, adenoidectomy. They're going to take out that obstruction and change it. And half of them were going to do watchful waiting because as the body grows, the change between the size of the tonsils or whatever is blocking it in the airway can change. And some of those children will then actually end up being able to open their airway and relieve their act. So the interesting part about this study, and they did a lot of measures for outcome, was that the way that we talk about severity of obstructive sleep apnea is an AHI, or an apnea hypopnea index. How many times an hour do you stop breathing? And so lots of these measures were indexed on people, especially when you're talking about pediatrics, that have nothing to do with the cohort that you're talking about, right? So AHI index and relating it to severity was normed on adult men. So it turns out that there wasn't any difference in cognitive outcome in these kids, whether or not they had a tonsillectomy or whether or not they watched and waited as per the AHI, right? So that index didn't really change anything for them. But what they did find in this study is that the depressive symptoms in particular were directly related to how low that oxygen went. So again, research into pediatrics is so important because many of the things that we take for granted that we know about health were not actually designed with children in mind. So this is just a reminder that the norms and the indices that we use are not translatable directly to kids. So not only does the AHI not work, what really came through here was not a cognitive impairment, which I think is what folks were looking for, but depressive symptoms. And again, it was related to the depth of oxygen desaturation. So hold your breath. So they're having the period of time, the apnea, and the oxygen is dropping as it's measured in the finger or the toe, right? And that measure was what was more closely related to behavioral symptoms. Okay. So that brings me to the other study that I want to talk to you about, present to you guys today. And this is what we call the POSA study. And it's a pediatric obstructive sleep apnea study, specifically looking at this phenomenon in children. So in children, what does apnea look like? Not just in the body as measured by the finger, but what's happening in the brain? And that's actually a really important and powerful question, because not everybody is going to have the same response to anything, right? And so what we want to know is which children are going to be able to protect their brain when we have an apnea, right? And which children are you going to see a drop in the peripheral bloodstream and a drop in the brain? Not every child will do that. Some children can cerebral protect, right? So you'll have a drop here, then the brain protects the oxygen supplied air. So not every kid is going to experience apnea the same way. And so this is a device study, and I've listed here for you guys to sort of read what we're talking about, which is what I've been talking to you about for about 5 minutes. That OSA is very common. The way that you make it in children, currently the AHI, the apnea hypopnea index, I told you about how we derive that from adult men, right? That's what we use, but we think it's probably more important to figure out which children can and cannot protect their brains. And here's a little, I don't know if my pointer is working, but for you guys, I can see it. Here's the little NIRS little flap that goes here, right? It's looking at what's happening in the brain. You have one on here that's looking at what's happening in the periphery. And then we validate that or run it alongside the polysomnogram. That's the gold standard. Because eventually what we'd want to be able to do would be to develop point of care diagnostics, right? So you'd be able to take this home, hook it up to a Bluetooth and record and see whether or not your apnea is apnea that we need to be concerned about. Okay. So this is a recap. I've talked about a lot of different things. This is the take home. And then on top of being a behavior, a function, and a process, you have primary sleep disorders that can interrupt the process, right? May or may not be behaviorally measurable, depending on if you're catching them, but that we think that definitely affects the function. Sleep needs are determined by homeostatic factors and developmental age, right? And again, process C and process S probably more greatly impacted by all those other sleep facilitators that we talked about, behavior, genetics, et cetera. The polysomnogram is a gold standard measure of sleep physiology, especially in children. But what we're working toward is being able to scale that up and get that out of the lab efficiently and accurately. And this, of course, is what we're talking about with apnea. There are other primary sleep disorders, nocturnal seizures, and periodic leg movements of sleep, things that fragment sleep. Anything that's fragmenting your sleep is likely interfering with the function of sleep, and neurodevelopmental windows in childhood are particularly vulnerable to those things. And that we use the sleep EEG recording as a way to image the brain, to reconstruct pathways, to understand what's being built. These are the studies. It's actually 2 studies that I have up here right now. So here's a QR code on each of them. I'm going to leave this up here. This is ESP with the rainbow pattern, and they're just 2 different flyers. And what they're saying is, you have a child who you want to be a research partner with us who doesn't have any developmental flags, or do you have a child who does have developmental flags? So again, 2 broad bins, which allows us to see many children. One category of children that's not included in ESP are children who have known and well-defined genetic syndromes. And I just want to give a teaser that we are actually developing and expect to be enrolling within the next 12 months, specifically for Down syndrome. So if that's something that you're interested in, or you know people who are interested in, please stay tuned for that. And then this one in the middle here with the QR code right here, this is the device study. So this is a broader age group, children 3 to 12. You are coming in once to see us. You don't have to have sleep apnea, because we want to know about what it looks like in children who don't have apnea. What is that cerebral protection reflex? What does that look like? And we do want to know in children who do have sleep apnea, particular different severities of sleep apnea, so that we can look at things like, how long are the apneas? Is that related to whether or not your brain can protect the drop in oxygen? How low is the oxygen saturation, right? To try to look at all those parameters. Again, 1 study visit, you get a physical exam. These are all in-house at the NIH. So I'm going to leave these up here, and I'm going to stop talking, because I definitely threw a lot at you, and just open it up for questions.
All right, Dr. Buckley, thank you so much. And would love for any of our participants today to go ahead and put your questions in the chat. So please let us know what questions you might have for Dr. Buckley. So a family's asking about cost, Dr. Buckley.
About cost?
Cost, yes.
Oh, so the wonderful thing about working at the NIH, and it makes my job as a physician and a researcher so fulfilling. There is no cost to these. This is, well, you can think about the cost of just like paying taxes, right? So the federal government runs this largest research hospital in the world, and the cost is your time, right? So we are looking for research partners, kind of what I said earlier. We can't answer these questions without the participation of all of you. So this should be thought of as a research partnership. If it's difficult for you to get to the NIH, transportation, things like that, we can work with families to help defray the cost, actually get you there. And there actually is a nominal reimbursement to you for your time. So it doesn't cost you anything. We can help you get there. And there is a reimbursement for your time.
Great, thank you so much. What other questions might you all have?
Okay. I'm looking in the chat myself.
Yep. Nothing
Okay.
yet.
So the sleep clinic is in Bethesda. I should say the sleep lab is in Bethesda on the intramural campus, right, I mean, like less than a mile from downtown Bethesda,
Okay.
between Wisconsin and Old Georgetown.
Okay. So not very far, certainly for our families here in Fairfax.
Not very far. Somebody has a question about the genetics of sleep apnea, and that's actually a really interesting note. So the risk factors that may make it more likely that you develop sleep apnea, probably genetic. So for instance, the shape of your face, whether or not your chin allows enough space for the airway when you're lying flat. People who have sort of very flat maxillary bones here. So there are some structural things that would make it more likely for you to have apnea. People who have, for whatever reason, if there's a genetic component to having very large tonsils as you're growing. There are two types of apnea in pediatric populations. One which we talked about, which is the mismatch between the airway and the tonsils. And the other one is children who have put on a lot of weight. So there's an obesity factor, and all children who have a lot of weight on should probably be evaluated. And then there's children with the mismatch in the airway. And the response to tonsillectomy, adenoidectomy, as you can probably imagine, is very good for this group, the group that has the mismatch, but it's not as good for children whose primary problem is weight. So it's not just the tonsils and adenoids, it's also the actual fat in the neck that makes it difficult to keep that airway open. So those are just examples of sort of the personalization of medicine. It's not a one size fits all.
Right, thank you. So we have another person asking if someone is interested, how long until actually, I guess, well, depending on what study this person is looking for, is it weeks or months away?
So right now we are doing the pilot for ESP, and we have 30 and change of the 50 total children who will participate in that. And so we are actively enrolling and getting those families in ASAP. So it's not months. It could be fitting in whatever your schedule is. It could be next week. So you would go through an intake. Our nurse practitioner, Jessica Vaughn, will call you, do a little bit of screening, and then they'll call you back. And through telehealth, you do a consent signing, and then you come in and see us. And so the day goes like this. You show up, you go through admissions, you get neurobehavioral testing with our sister group who does all of that. If you've decided that you want to be a part of the genetics, you'll meet with the genetics counselor. And then in the afternoon, you put your leads on. And then plus or minus the blood draw then could also happen the next morning. And then you go to sleep in the lab. In the morning, you wake up and you go home. So we can turn that around very quickly. It just has to do with, what's being booked in the lab, but certainly not months.
Okay.
Yeah.
So
And
Dr.
then,
Buckley, I'm sorry, for the ESP study,
yes.
that you really are looking for children, though, ages 3 to 6. I just want to make sure that we're clear about that.
Yes, that study at the moment is the younger kids, 3 to 6. We're enrolling the next 20 of them. The device study, which does not have a blood draw and does not have behavioral testing, is 3 to 12. Okay. And so if you suspect that your child may have apnea, and they're a little older, that's a great study to come in and do. It's one time you come in, you do the device alongside the PSG. If you don't, and you'd just like to be a research partner, you don't suspect your child has apnea, we would love to see you as well. So yes, that study is a little broader.
Okay, thank you. So another family is asking about, can children diagnosed with autism spectrum disorder participate?
Absolutely. And we do have a number of children who have ASD. Yes.
Okay.
Great question. You don't just have to be sort of suspected. You could have a diagnosis of ASD. Absolutely.
Okay. Would you all eventually have a study on older kids, like 10 to 12-year-olds?
Yeah. So right now, again, the device study is open for children 3 to 12. So it was originally written for 3 to 8. We just expanded it because there is so much interest out there. And I would absolutely love to expand further from 12 to 18. And that is the idea, right? These things happen incrementally, and as there's interest, we expand them. So at the moment, if your child is 10, you would qualify for, or may qualify for the device study, which is 3 to 12 years. And then hopefully in the very near future, we'll broaden that out.
Okay. And so what type of data results could one expect to receive from the device study?
Great question. So because the device study is being validated against gold standard, which is the polysomnogram, you will get a full polysomnographic report like you would if you got a sleep study somewhere else, right? So we would tell you about the number of cycles, all your latencies, whether or not you have any evidence of movement disorders, whether or not you have nocturnal seizures, and then most importantly, whether or not you have obstructive sleep apnea, because that's the most common thing that we would see.
So, Dr. Buckley, if there's some type of concern as a result of these studies,
Yes.
how does your team support families
Yeah.
in kind of moving forward and understanding what interventions or supports they
Yeah.
might need?
Yeah, that is a fantastic, wonderful question. We do a lot more of this. Our lab is pretty much built with the idea that that is actually what, these are very young children, right? People sometimes are coming in because they have concerns. So we have that happen all the time. We may be the first person to tell you, we found what looks potentially like a nocturnal seizure. And in that case, and I'm a pediatrician, right, at heart. And in that case, what we do is we make very clear that we are a research team. We are very good at what we do. We can give you the diagnostic information, and then we facilitate with you somebody who is not research teams. Therefore, they don't have any conflict of interest for taking care of your child, somebody who you can see as a specialist to evaluate what we found. And so we do a very careful and concerted handoff, and whoever you find we can give you referrals if you don't have that person, or if it's your pediatrician or your neurologist, we speak with them directly and tell them what our recommendations might be, but with the clear understanding that the clinical management of that person needs to be done inside the research environment. And so that's what we do. We give you back anything that we find concerning, and then we facilitate the handoff to someone who can take care of them.
No, thank you for that, because I'm sure some of our families were wondering that, because
Yes.
it's hard to get information and then not know what to do with it.
Oh,
So
we never leave people hanging like that. And I will also say that even with an abnormal result, that doesn't necessarily disqualify you from the study. So then we can continue to follow you along with your
Okay.
outside team. And so you get hands and eyes on in that way too.
No, that's great. Thank you. And another, when the family member who was asking about, for older children and would
Yeah.
that eventually come along, I think they raised that question with regard to just concerns around kids who are on screens late or gaming, and how that might affect what's happening with their sleep patterns. So I know there's a lot of research out there around all those things and screens, but I think that was just a point that they were making, that, um, how is that really impacting our children's sleep? And I know we have a lot of families who are asking those questions.
Yeah. And that is probably a much broader,
Right.
a much broader question. Remember, to the
Yeah.
process C and process S, and then everything underneath of it,
Right.
right? All of those things that then impact actually whether or not those things are lined up the way that they should be. And certainly screen time or staying up at night because you're fixated on the very addictive properties of social media, certainly. I think certainly that affects it. I know there are, just as an interesting aside, there are states that are taking measures, laws, to sort of put the brakes on those things. I think Utah just passed one. California has a different one. So it is certainly topic of conversation, but not one that I'm an expert in, other than the parent.
No, absolutely understood. So another question. So what days of the week are the actual studies conducted? Are there any limitations?
Great question. We have a lab that runs Monday through Friday. So the only limitation is at the moment, we don't do Saturday, Sunday studies. So you would probably, for the device study, maybe come in the late afternoon, 3 or 4. For the ESP study, you'd be coming in the morning because we're doing the behavioral battery as well and potentially meeting with genetics, etc. But other than that, no. The only thing I should mention is that children would need to be accompanied by a parent. So one parent must be in the room overnight with the child during the sleep study. That's the rules of the lab. And other than that, no.
Do you, I'm just very curious, with the younger children, with 3 and 4-year-olds, how difficult is it to conduct that overnight study with
Oh,
them? Because
yeah,
I'm just thinking,
yeah,
when mine was that age, that might have been a really tough thing to
yeah,
do. How does that look
yeah.
in your lab?
I get that question so often, and I will preface the answer by saying that we've done close to 1,000 pediatric sleep
Mhm.
studies at the NIH, right? So
Yep.
our team is amazing. So the group that I showed you in the very beginning,
Mhm.
there are a number of those folks who are nighttime sleep technologists, including our head technologist was in that photo. And these people have been at the NIH for 2 decades, like 2 of them longer than I've been there. And they have put leads on and conducted sleep studies with any and all type of children that you could imagine. And so part of, doing the study like this is having a great pediatric team, having a team that understands children. So for children who do not have sensitivities to their to having their head touched, it's a very easy thing, right? You speak to them, you tell them what you're going to do. It doesn't hurt. It's completely painless.
Right.
And you watch a movie or you read a book or you do what you need to do. For children who have more tactile defensiveness, it takes a little
Mhm.
bit longer to gain trust, but that's what we do. So we have done
Okay.
many, 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 many of these.
No, thank you for that. I just was thinking there might be some families out there who are
Yeah.
maybe a little concerned about that.
Yeah,
All
and we would,
right.
and if you call and speak to Jessica, she will
Yeah.
go through with you any of those paradigms that we use.
Okay. Um, so another question. So single parent who has 2 children,
Yep.
is that a possible scenario to have one child studied? So I guess I'm not quite sure.
Yes,
Yeah.
I think you
Okay.
might be asking, are you talking about the difficulty of coming in and having to spend
May,
the night? Okay, yeah,
maybe. Yeah.
yeah. If that's what you're asking, and I totally understand, I totally get it. We have to have a parent overnight in the room with
Yeah.
the child. We can't get around that.
Yeah.
And the other child can't be overnight.
Right.
So
Okay.
yeah, we
Yeah.
don't have a solution for that at the moment, and that's kind of out of our hands.
Okay. No, thank you. I appreciate that. Any other last questions for Dr. Buckley? So Dr. Buckley, I want to just give you the last word.
Sure.
Just around uh, for our families and just
Yes.
what they might be able to
Yes.
do around this. So
Uh, I
your
can
final
only
word.
thank you. I can only say that I think you all know, for those of you who tuned in today, that sleep really has to be thought of like food and shelter. It is actually really
Mhm.
important. So there's that, right? There's the whole preventative, thing about protecting sleep because it needs to be protected. And then there's the mysteries of sleep. And we really think that our approach is going to allow us to identify behavioral trajectories that are problematic for children and families well before that behavior surfaces. And so, in order to do this research, we rely on partners. We rely on you guys to be interested and faithful colleagues with us, really. And so this is an appeal for you to click on that QR code and find out a little bit more about what we're doing. And if any of these studies in particular don't work for you, keep us in mind. And we are a very busy lab.
All right. Well, thank you so much, Dr. Buckley. Thank you, everyone, for joining us today. And if you have any other follow-up questions, please let us know. We can certainly get you more information as needed. But thanks again, Dr. Buckley. Wonderful information and good luck with the studies.
Thank you. Bye all.